So we are picking up chapter 5 just after the first exam. This is material that will be on the second exam. It's the end of chapter 5. Two guys on a little bitty island. One of them says, I wouldn't worry. With continental drift, Africa or South America should come by eventually. That will make sense before we're done with the chapter. Or at least you'll know what the connection is. Lamarck did not know how things were passed on, right? Inheritance of acquired characteristics. He knew things were passed on, he just didn't know the patterns that they followed. But he knew it was important that features that were useful in an environment would give their possessors survival abilities and they would pass them on. Now, when Darwin came along, people still didn't really know how stuff was passed along. But between the time Darwin first published his big book and second or third edition, the idea of inheritance of acquired characteristics had kind of gone away because there's too many instances where it just didn't make sense. But nobody still knew about the stuff we've talked about in this chapter. All the build of genetics from Mendel to Watson and Crick to how DNA actually works. On the molecular level, we cover that in the second semester, but now we're going to talk a little bit more about once genetics was kind of out there, the patterns were recognized, it started to be folded into evolutionary theory. And what they called this was the modern synthesis. Darwin talked about changes in populations, changes in groups of individuals. People started to realize that that group was not just a group of individual organisms, it was a group of various alleles. In the same way that there was variation among the individuals, there was variation among their genetics. They were a big mass, a big pile of alleles. Some alleles common, some alleles rare. But the alleles were connected to producing the different traits that evolution is kind of predicated on, right? Conditions change, some traits become advantageous that weren't, some become disadvantageous that used to be good, but now we're looking at what's producing the traits. We're looking at allele ratios. So conditions change and one feature becomes advantageous. In order for it to work evolutionarily, it needs to be inheritable. In order to be inheritable, it's connected to certain allele ratios. And that's what's going to shift, is alleles for advantageous features are going to become more common in the survivors. Well, the survivors are more likely going to have them. Their offspring are more likely going to have them. Other alleles are going to come in from maybe other survivors who have a different combination of good alleles, and then the offspring start to get all these great mixes of alleles. And so that's kind of the genetic version of evolution. Sorry. not a huge shift. Darwin would say in any population there are variations amongst the individuals. We can say in any gene pool there's variation among the alleles, and alleles are possessed by different individuals. Those combinations that are good produce more survival, produce more offspring, become more common. People started to understand what these actual reproduction types are, right? Asexual reproduction, copying and sending on the same genome, right? Offspring are genetic copies of the parents. Asexual reproduction, mixing. Everybody's got a genome that has two copies of everything. Two, maybe the same alleles, but two alleles for the same gene, and so you get variation there. And when you mix them, you get all sorts of other variation. So in asexual reproduction, how do you get new alleles? You get them by changes in the DNA, by mutations. And that can, over time, with many offspring, many instances of copying, and copying that can go wrong, can produce variation in the population. I also have, in parentheses, turns out that there's a lot of tricks involved too, is that bacteria will actually package interesting alleles, useful alleles, in little things called plasmids, little loops of DNA that they can then share around, not just with bacteria of their own species, but with other bacteria. Sometimes even we have bacterial genes integrated into our genome, most of which we don't use because they're bacterial genes and they're less useful than problematic, but we do have some genes integrated from bacteria, you can see from the sequence, that we actually do use. Is that, if I'm remembering right, I read this earlier in the year, a major player gene in our immune system function is traceable to an original bacterial gene that in this sharing thing we probably wound up with. And that's what happens. They share these little loops. They get integrated into other cells. They don't just use them. They wind up integrating them into their own major copy DNA and then passing it on to their offspring. So even they have what you might call quasi-sexual reproduction, another way of producing variation other than mutation. But of course, in sexual reproduction, every time you're making offspring, you wind up mixing a set of alleles with a set of alleles from another source. Maybe it's a source within the same individual, maybe it's a source from another individual, maybe there's males and females involved, maybe not. That's a whole other thing. Now, one interesting thing in the world is there are organisms that do both. Oops. And it's not just a situation where they can do both. There's a number of single-celled protozoans that can do sexual reproduction or asexual reproduction. It kind of depends on the circumstances. A lot of funguses can do that also. Sexual reproduction under some circumstances, asexual under other circumstances. But there are some species of plants and some species of animals where this is baked in. every lifetime has an asexual stage and a sexual stage, and then an asexual stage and then a sexual stage, and another lifetime, an asexual stage and a sexual stage. Now you look at that and go, why? Well, think about this. While you're adapting, your population is trying to deal with conditions, every time you get a good mix, you can make copies of them. You get a good mix sexually, then you can make copies of them asexually. And now you've got a broader population of a good mix. And then the next stage is to mix it again and sort and mix and sort. The amazing thing to me is that this is not a common thing. You find this in land plants. We'll talk about this later in the semester. When land plants first invaded the land, they still had not lost the need for open water for the sperm to swim to the egg cells. And so they developed an alternation of generations approach where the sexual stage was very small and on surfaces that could accumulate water, and the sperm could swim to the eggs. And then the sexual stage became a bigger stage that could produce sexually copied airborne spores to spread the plants across the land. Parasites, this is also a common approach, where a parasite will do an asexual stage in one host and a sexual stage in another host. Malaria does the asexual stage in humans and the sexual stage in mosquitoes. There's a bunch of worms that do this as well. So when you do find this as an approach, there tend to be very specific things kind of go along with it. Parasites, early land plants. Some things like corals, where the major form that you find them in is kind of stuck in one place. It doesn't move, right? Corals build this rocky stuff around the living corals. They can't really move with that. And so that becomes their asexual stage. They just make copies and grow the coral, make copies and grow the coral. But to spread the coral, they produce, asexually they make genetic copies, but the genetic copies use different genes to be little swimmy jellyfish. And they can spread, reproduce sexually, no male and female there, well, sometimes male and female there. And then the larvae, if they're lucky, they settle someplace close to the surface, new islands rise up, whatever, and they can start a new coral colony somewhere else. So those are the basic ways that alternation of generations works. Another thing connected to this, something we did talk a little bit about earlier, molecular clocks. By far the most common mutation are point mutations, single changes in the sequence of DNA. If you take species A and species B, you know they're related, but they're different species. And the question is, so how far back on their family tree did they branch out from a single starting population? And the idea of molecular clocks is point mutations accumulate with a regularity, tick, 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 tick of a clock. So long as you look someplace in the genome that doesn't seem to be particularly prone to selection. Like, you don't look at genetic sequences because alleles vary and alleles are selected for. But there's a whole bunch of DNA, they used to call it junk DNA. And even if it's functional, changing it with point mutations doesn't seem to mess anything up. So you compare parts of the DNA that are this supposedly non-coding, non-functional DNA, because it can afford to change over time. And I'm making these numbers up. If a point mutation occurs every 10 generations, and you look at your two species along a stretch, and you find 5 point mutations here that don't match 5 point mutations over there, then you're talking 50 generations, 5 times 10 generations for each change that happened on one branch that didn't happen on the other branch. It's uniformitarianism. How do we come up with the clock? Well, we take a look at how often mutations happen, and we kind of assume they've always happened at those rates, which is, silly on a certain level, because mutations are affected by, the environment, chemicals, radiation, that sort of thing. But we're kind of stuck with it because, as uniformitarian is, we don't know what else to do to apply to this thing back in the past. But you will find where the radioactive decay analysis determining the age of fossils, that's one way of looking at how old an ancestor is. And then you've got the molecular clocks looking, and the people that do this, if their numbers don't match up with the radioactivity stuff, which is a lot more reliable, still uniformitarianism, but a lot more reliable, they'll insist that they're right and the radioactive stuff is wrong. Because that's the way people work, right? Our numbers are better than your numbers. This brings us to Hardy-Weinberg. Hardy and Weinberg were statisticians. And people were discovering situations where conditions would change, and within the gene pool, there would already be alleles in place that turned out to be very advantageous for the new conditions. Kind of sort of look like Lamarck's idea that there's a plan and you get what you need when you need it. But people went, well, that doesn't work. But if this unusual allele in the population had appeared as a mutation, isn't it going to just, it's a tiny fraction of the population, isn't it just going to fritter away? Is that it's not going to hang around for 15, 30 generations until it's useful, right? Well, Hardy and Weinberg went, is that once you get an allele in a population, in a gene pool, as long as certain conditions are true, it will stick around, it will occupy the same ratio within the population over time. So as the population grows, the number of possessors of that allele will grow, right? You get a mutation in you produce 5 kids, 3 of them pick that mutation up. Now it's a little bit more individuals have it, but the population has grown too, so the proportion stays the same. Now, when I first learned about this, you learn about it and you go, this works if under these conditions. And I remember, as a sophomore in high school looking at that and going, but those conditions don't exist in the real world. What the heck does this mean? And it turns out that what this applies to very often is told populations have to do this. Well, no, they don't. Is that this applies to sometimes to the population, but a lot of times to the particular alleles. And when you do apply it to the alleles, it makes more sense. So you get a new allele, and it can produce a particular change in a feature, but under current conditions, it isn't positive or negative. It doesn't give you a natural selection, a survival advantage or disadvantage. It doesn't give you a reproductive advantage or disadvantage, sexual selection. And as long as that's true, it can hang around at a certain proportion within the population indefinitely. How'd the allele get there? It was produced by a mutation. possibly brought in from another species. That happens too. But what are the odds of it changing again in a significant way? Well, the odds are pretty lousy. So you can say, there's no selection on it, it isn't itself going to change again. That'll keep it around. If it changes again, it's a different allele. But the odds are, when you're talking about mutations that occur very, very, very rarely across one spot on the genome, across the whole genome, there's lots of mutations that happen. But there's billions of spots on even a small genome for a mutation to occur. So the odds of it happening twice in the same spot are pretty low. Now this one has to do with the math. If a fragment of the population leaves, it leaves the main population and sets up shop somewhere else, then it's going to change the ratios of the whole population, because it's going to take some stuff with it, just kind of randomly. It's going to take some stuff with it that lowers the ratio in the remaining population, and it raises the ratio in the little sliver that's leaving. And Harvey Weinberg were all about allele ratios staying stable. We're going to have a couple of features that connect to this when we get done with our little list here. But migration into and both out of and into a subpopulation that's been split off, doing its own little cooking somewhere else, now joins the main population for one reason or another, bringing its alleles in. That's going to change the ratios as well. So an allele arises by mutation. Say I'm carrying an allele that was in a mutation of me as an embryo, first cell. Got it maybe from the sperm or the egg. And 50 generations down the line, if conditions change, that particular allele, for whatever variation it produces in me, turns out to be very advantageous under those new conditions. But before I ever made offspring, something bad happened to me. Tree fell over on me. It's nothing having to do with the allele, but bad luck. That's a numbers problem. It's the same reason you don't run an experiment on a very limited number of subjects, is because the odds on a small group just screw a lot of things up. Just chance does. But say I pass my allele on to my family and they pass it on to their family, and they stay the same proportion of the population. But that proportion is eventually a lot of individuals in a big population. Now the odds of a tree falling on all of us is pretty low. And so what this does is, just like it does when you're designing an experiment, a large population removes the element of chance at changing the ratios of the population. Here's some, now that's the list. Here's some stuff that goes with that. Okay. If you look at the population of Native Americans, you look at blood types. Turns out type A, which is a genetic, inheritable feature, is pretty rare in Native Americans. And the question is why? Is it bad for them in some way and it's been kind of weeded out? It's probably a founder effect. The Native Americans derive from Eastern Asians. So you go, oh, is it really rare in Eastern Asians? It is, but it's not as rare as it is in Native Americans. But the splinter group, the subgroup that migrated from Asia to the Americas, actually on screen, I should be going that way, I guess, was only a few hundred people. Very small population, right? Not a huge population, but very small population. And so they're working their way along the sides of the oceans, probably in small boats, canoes and stuff. And so you've got maybe, we'll say 30 or 40 families doing the early migrations. One of the canoes goes down. That family drowns. Small group, they may have been most of the possessors of A alleles, blood type A alleles in that population. Another chance occurrence, nothing about it made it likely that they were going to drown. But now not only was maybe the population depleted in A alleles in the first place, but now it's lost a huge fraction of those A alleles. But it's going to go on to found the whole population of North American and South American natives, mostly. So they're stuck with that starting allele ratio of not a lot of A alleles for the population. It's a founder effect. The subgroup is a founding population, and the descendants are stuck with that starting allele ratio for the most part. Bottleneck is a similar situation where you wind up with a small group stuck with the allele combinations in the small group, but instead of a migrant group, you get an almost extinction event. Population gets reduced to just a small group. And whatever alleles are in that group, that's what everybody's going to be building on for the future. This happened to us. I forget, 3-4 million years ago. I forget. I get the numbers wrong. But a long time ago is that human ancestors originated in Africa and then spread out across the world. They wound up in Asia, they wound up in Southeast Asia, in the islands. They wound up at least partially in the middle of Eurasia, maybe into Europe, definitely across the rest of Africa. Something happened. And those populations of humans died off. Virtually all the populations of humans died off except for a cluster still in Africa, who, after a while, did it again. But the groups that had spread out originally, whatever allele combinations adapted them to those new populations, because what you get with a mutation is random. If that near extinction had never happened, and the spread human populations had continued to adapt to the different areas and evolve, then the racial, I can't think of the word, but the racial distribution, the features of all the different races would probably be very, very different. You'd have some stuff that's kind of predictable, like skin color, because skin color is associated with sun exposure, but other kinds of things like fat distribution in the faces that changes some of the appearances of some races, is that those are all things that probably were not terribly adaptive. And maybe those changes don't appear if we hadn't all shrunk down to the bottleneck and come back up. Is it? So again, small population, more chancy stuff, that sort of thing. The third feature that we'll talk about before we stop this particular video is genetic drift. Once you do have separated groups, which is what we're going to talk about in the last part of this chapter, then the mutations that occur in one are different than the mutations that are going to occur in the other one. And so even if they have separated into very, very similar conditions, you may see differences enough to make them clearly different species in these two different areas. If the conditions are really different, then obviously there's going to be selection and you're going to see differences. But some of the differences between leopards in Africa and jaguars in South America, slightly differences in color and how the spots work, probably are just a product of genetic drift. is that overall some features have just kind of shifted away from the other population, and enough to make them, clearly different, as jaguars and leopards are pretty easily distinguishable from one another, even though they're very similar big spotted cats. And they have very similar lifestyles. They live in the tropics. Jaguars are more rainforest than leopards. That might explain some of the differences. But a lot of it comes from just the drift. Is that human races, some of the difference is probably just genetic drift. It isn't adaptive in a particular set of circumstances. It's just that population got some mutations, and for whatever reason, those mutations became more common. There is something, it doesn't really have a term. Sometimes what happens is a new allele will piggyback on an existing really useful allele because they're linked very closely together on the same chromosome. So it's always being sent on as a package to offspring. So you send a really useful allele piggybacking, not really useful, maybe even slightly, useless allele with it. And that sometimes is the pattern that you can find in how stuff spreads through a gene pool over time.